Now, is that good? Okay. Can everyone hear me from the way back there? And see the slide presentation? I hope that all of you can. I received much too much adulation that's really good for my own good. but I'm very, very fortunate. Yeah, I'm going to tell you a little bit about my story. I'm an atomic physicist, and when I was a small boy, I always loved the truth. One of the ways that I understood to learn the truth, I learned that the scientific method could help me understand what's really true. So I became interested in science, and at that point, I would study plants on my own. I was a very strange kind of kid. So I would cut a piece of a leaf or a piece of grass and watch it grow back in perfect form and structure and say, my goodness, how does it know how to do that? You know, and as you start studying science, well, things get smaller and smaller. First of all, you start studying tissues and structures, then you start studying cells, and then you realize that the world that is inside of cells is absolutely fascinating. And you have to go inside the cells and understand what the molecules are doing, and then you go inside the molecules and understand how the atoms fit together. I figured that I could understand the whole mystery of life if I understood how these little atoms could work. And it fascinated me so much that I actually went in and studied atomic physics and learned all the quantum mechanical effects and et cetera that made atoms fit together into molecules. And that was really exciting for me until I realized that we really don't understand even a very small fraction of what is really going on. And so I decided that after I graduated with my PhD that I would go into a field called nanotechnology. Nanotechnology has to do with very, very small things that are the size of molecules, of large molecules, only 200 or so atoms big. And we could make particles that could go inside living organisms and do interesting things, such as disable viruses. We found particles smaller than a virus that could hook onto viruses and then disable them. We found particles that would be able to accumulate inside cancerous tumors. I can actually say that in terms of nanotechnology. Then deliver directly the things that are needed to kill a tumor, or we found particles also that could be tuned to certain microwave frequencies that would heat up the tumor and cook it. It was really, really interesting stuff. But in the process of getting these technologies out to people, we realized that there are quite a few hurdles, and most of them are financial. And a great deal of work has to go on in order to bring these technologies out into the public realm. So now I get to this point in my life, and I've spent quite a great deal of my life studying science. And I met this gentleman, Vertus Norton. He's my neighbor. He lived about a mile up the street. He heard that I was an atomic physicist and asked to go to lunch with me. Well, I was involved in nanotechnology, and I was interested in being able to commercialize these technologies. So I put some bottles of nanoparticles in my pocket, went to lunch with Vertus Norton, and you can imagine what I was trying to do at that point. He was a Fortune 50 businessman, had reorganized and restructured strategically $30 billion industries. And I said, wow, this is my chance. So I sat down to lunch with Vertus Norton, and lo and behold, after explaining all the wonderful technologies that we could do, we had stuff that could cure malaria. It was just fantastic. And he said, Gary, I'd like you to take a look at this. And he put on top of the table, alongside my nanoparticles, a bottle of salt water. And he said, I want you to take a look at this. And I said, well, what is it? He said, it's made out of salt water. It does some really remarkable things. It's helped me with my health. And I said, oh, my goodness. Why does an intelligent, extremely adept man have such a fixation on salt water? I didn't understand it. But I thought well enough of him to be able to, say, well, maybe I better guide him a little bit. And so when he said, Gary, would you help us figure this out? I said, oh, of course. I learned that there are about 16 years of studies, many millions of dollars worth of studies that had gone into this. He dropped it off at my house in big boxes, and I during the evenings I had time to look at them. There's a few things that I realized right off the bat. One is that in over 16 years of toxicity studies, these are the same FDA-required studies that are required for any type of pharmaceutical, that there had been not one indication of toxicity found. Not even in one animal or one case. And so I thought, well, this is very strange. I mean, this is not something that we see every day. And so I said, well, if there is no toxicity found, if it does anything beneficial, then we're ahead of the game. So now I'm starting to turn around a little bit and saying, well, maybe there's a possibility here. Then I read through more of the studies and realized that it had very beneficial effects on animals in different situations. I thought, my goodness, well, we have probably a very promising technology. One of the things that I was impressed with is that this technology, this ASEA, has some of the most reactive components that ever have existed on Earth. They're bioactive. And you'd expect almost immediate response to cells, a toxic response. And yet with all of this, these studies and evidence, there was absolutely none. And so I came and said to Virtus, this is impossible. It truly is impossible. I don't see how anything could be so bioactive and yet have absolutely zero toxicity. In fact, these are the same molecules that the immune system uses to kill bacteria and viruses, the exact same ones. And they have enzymes and such in the body that help to regulate and break them down. And just to be able to see that in a little window of a possibility here was exciting for me. Because, let me just say one thing. All scientific discovery happens because someone observed something and said, why does it work? You think about that for a minute. And so we found that penicillin kills bacteria and yet is not healthy, but is it tolerable? And that's wonderful. And we've saved millions of lives because of that fact. Something that is non-toxic and yet bioactive. And then I thought we really ought to look into this. In fact, we have a moral obligation to do so, so I had to kind of crawl back to Virtus and tell him, well, Virtus, I think this is incredible. This is something that needs to be studied, looked at, and Virtus said, with a smile on his face, will you help us figure it out? So we went and met with a pharmaceutical company when we put all of this together. You've heard the story. And then realized that, and actually I figured it for a while, that if this went to a pharmaceutical track, there would be maybe one indication, one effective indication. The rest of it would be thrown away. Possibly everything would be thrown away. And nothing would come to the people that needed it. And that was a changing point for us. And I remember around the table as we were discussing this, and I kicked Virtus under the table and said, you can't sell this. We can't let this fall into hands to these type of things, where only financial interest is at heart. So we had figured. And so they decided to turn it into a direct marketing company. And I said, Virtus, do you really have to? And he said, Gary, it's the only way. We can't get it out to people otherwise. And, I was saying, well, can't we just something else? I remembered that it was I remember thinking, this is the end of my career. I can't walk away from this technology. It is doing too many wonderful things, and I've seen people come up to my own house and thank me hundreds of times over for the things that it was doing. You cannot ignore that. So anyway, I'll tell you a little bit of the journey of discovery that we had to find out this kind of story of how I came about the understanding this is real, if you will. This is a Yosemite National Park. Probably some of you have been there. It actually exists. If you haven't been there, you'll just have to believe me. It's beautiful. We've mapped about most of the country. There are some areas, though, that haven't been mapped. Now, scientific truths are a lot like physical geography. They're just true. They are because they are. And all we have to do is to be able to go and discover these things. Well, in the frontiers of science, this is kind of what it looks like. You're walking along a trail, maybe only 100 or 2 have been walking along this trail in the past. And then you come across a trail that kind of leads off to nowhere. And you ask yourself the question, where does it lead? Where does it go to? And what we usually expect to discover is about something like this. It's just kind of more of the same. Nothing that hasn't really been discovered, hasn't been discovered in the past. What we found with ASEA is this technology has almost unlimited potential. The horizons go on forever. And that was very exciting for me. Some people have done research on these types of molecules, and we're going to be talking about the types of molecules that I mean in a minute. But very few have really made great inroads. We found a way that could be of great benefit for the discovery of many more things. Let me talk about scientific discovery. About 1665, there was only one journal that has been published. One. And for about 100 years, that was it. In the 1900s, that's only 5 generations ago, there was 250 journals, and people like Einstein, Max Planck were publishing. In this generation, there's 23,000 journals being published and 1.4 million articles. And that accounts for 10 million months of science being published every month.
Wow.
Can you imagine the pace at which scientific development is capable? Our patent offices, our infrastructure to assimilate and get this science out to people is so inadequate, in consideration of how much we have coming forward. This redox biochemistry field is also progressing quite nicely. In 1940, we realized that the cell structure and mitochondrial function. In the 1970s, this is my generation, we realized the importance of DNA structure and protein synthesis inside the cells. And now we're starting to realize that DNA isn't everything. In fact, DNA is only the computer program, the messengers, the epigenetics that carry the messages from cell to cell are the things that push the genetic buttons and make the cell act as it does. And so now it's epigenetics that is becoming more of interest to science. What is happening in these messenger systems? And many Nobel Prizes now have been gained because of research now in this field. Redox biochemistry and redox signaling being some of the foremost in this field. Here are some people, for example, that realized that this very simple nitric oxide molecule had very significant effects on cardiovascular function. they won a Nobel Prize. Here are a few that realized that certain protein structures, called G protein, are very influential in how the cell communicates. And here's a guy, James Watson. He discovered co-discovered the helical structure of DNA, very well known. And he has said that probably the most important work since the double helix is the role of reactive oxygen species as both an elixir of life and a deadly force against stressed cells. ROS, by the way, is the scientific code name for what we call redox signaling molecules. Okay, so now some of the greatest scientists on Earth are starting to realize that there are very essential roles being played inside of our body, these signaling molecules. And these oxidants are not all bad. I'm going to go back into this a little bit. They actually do things inside our body that are extremely important, fundamental. This guy, it's kind of interesting. I think he wants to go for another Nobel Prize. And so he's championing the cause of redox signaling and these ROS molecules. He says that if I'd like to find multi-billion-dollar funding for the production of ROS by exercise, because you can exercise and create it inside your own body, and see how that works. Anyway, how about this field of redox signaling? It has to do about the diverse and complex structures of life. I'd love to do this. Look at your own hand. All of the joints and connective tissues and functionality. If you were just to study microscopically the surface of your hand, it would take maybe 2 or 3 weeks to figure out what's going on there. And that's nothing to say about what's happening beneath the tissues and all of the wonderful structure and of your hand. All of this is made out of interconnected tissues. Tissues are made out of interconnected cells. And cells, like I mentioned before, are made out of interconnected molecules. And these very complex molecules, like these are the G proteins, and carry on the myriad processes of life inside the body. But what connects all of the molecules? Water and salt. Literally, salt and water is the medium of biology. Everything all biochemistry takes place in water and salt. And these redox signaling molecules in saltwater medium basically form the medium by which all of these are able to communicate. Now, I play the violin. Not very well. I won't play it for you. Don't worry. But when you realize... Hello? See, that was a wake. I programmed that to wake everybody up once in a while. You start realizing that if you want to really play good music, you've got to listen to the people that are next to you. When you start feeling what their part is and your part is and how the interactions take place, you can really make some beautiful music. The harmonious orchestration of life is very similar. The interconnected molecular players perform their functions when they're able to get signals and listen to one another. So who or what conducts the orchestra? I'm not going to go into that. But the interconnecting ocean of vital fluids, if you look at things on the molecular scale, a drop of water has 1 billion water molecules. One drop of water. And so you can imagine that there's a virtual ocean when you're looking at a molecular scale of this water surrounding all of these biological molecules in your cells. It's kind of fun to go there once in a while and swim around with the molecules. But the science of redox signaling has been developed quite a bit during the last little while. With this understanding, Britton Chance laid the foundation of that oxidants are beneficial signaling molecules. So when we thought of oxidants as being evil and we need to get rid of them, he starts saying there are some fundamental roles these oxidants play inside of our body. We ought to study these. Dr. Ruma Banerjee put together a beautiful book on redox biochemistry, which explains, it actually compiles some of the papers that Dr. Carl was talking about earlier. There are books, journals, and conferences all over the world now on redox signaling. The funny thing is that most of these people don't even realize that it's possible to stabilize some of these molecules. The basic laws in biophysics of life are such as this. I'm going to go through this again. Oxygen, nutrients, hydration, and energy go inside our body, are digested and get inside our cells, and create activity and energy. And inside our cell, then, is where all of this interesting activity takes place, where the fuels that we eat and digest, as well as the structures, are digested and put inside the cell, and there they do their functions. Molecular machines interact by altering the redox potential inside the saltwater medium. I'll explain a bit, but not too much. Redox signaling mediates all the communication and coordinates the activities of these molecules inside the cells. So we're talking about some very fundamental science here in cellular biology. So they help create energy. In fact, they're produced, like Carl said, in the mitochondria by a process of metabolism of sugars to produce ATP. And they aid in the damage detection, repair, and immune response, and much more. And create, as sort of a byproduct, these redox signaling molecules, or ROS, as the scientists call them. And these redox signaling molecules then perform the basic functions inside the cell. Here's some of the molecules, by the way. We'll talk a little bit about those. What we've been able to do then is take the same combination of molecules that are produced inside the cell by the mitochondria, and then stabilize them to a point where we could bring them inside of a bottle. And that was my work. It took me about a year to figure out how to do this. So, thank you. And now it's taken me many more years to tell people about it. but anyway, here's sort of the process by which this happens. We take salt, which is sodium chloride, like you said, just pure salt. No minerals or anything in there, and water. And it goes through a process of reduction and oxidation to produce some very active molecules: hydrogen peroxide, oxygen, superoxides, hypochlorites.
Dr. Samuelson, I really want to see this. If you could just shift around a little bit so I can see how
It's a secret. I can't tell anybody. Is that better? Oh, I'm sorry. I apologize for those people over there. It's good to see you, John. So the combination of these molecules here, or recombination of the molecules, I say, form these more than 15 types of molecules. Now, I love Carl, but he's not an atomic physicist. There really isn't an 8 and 8 mix. These things kind of recombine into different types of molecules, but the redox potentials, there's both oxidative and redox potentials. And all of these things are perfectly balanced to the point where if you were to recombine them or take them apart, they would form salt water again. So does that make sense? No, it doesn't really make sense. Okay. Okay, well, I'm not going to try to go into it, but what I'm trying to say is that these things are so native to the body, or natural to the body, that when you put them inside a living organism, the organism knows exactly what to do with them. And that's pretty neat, because then that also accounts for the reason why they're not toxic inside of our body, because they're already there. The body knows how to use them. It's kind of funny because hydrogen peroxide itself is a known toxin. Some of these superoxides, by the way, you said that they only exist seconds. Actually, they only exist millions of seconds inside the cell, and are very unstable. These molecules are interesting, but they are so reactive that they don't stick around. So we asked the question, well, what type of chlorine species are inside this? We can answer this by a multi-million-dollar NMR spectroscopy. And this deals with what kind of chlorine compounds are in there. We looked at salt water as a comparison, and sodium chloride as a comparison. ASEA actually was neither salt water or sodium chloride. It kind of had a different composition stuck in the middle of those two. So it's a different type of complex. And it's a combination of some of these molecules. Not exactly 8 and 8, but they're complexes that form from these. Also we have the superoxides, which I mentioned are just very fleeting. They don't exist for very much longer than a few millionths of a second inside a cell. And inside this product, we've seen that they are preserved for years. And this, by the way, if I could publish, and I would love to be able to publish this maybe later on, we'll be able to see that we figured out a way to stabilize superoxides. And so the immune cells use these things. This is oxygen, superoxides, hydrogen peroxide, these things that we've been talking about. The immune cells use these things as they surround the bacteria and kill it. These are antimicrobial. And I'm sorry, I shouldn't have said that word. So strike that. They're very effective against certain little things that happen in your body. All right. Besides being antimicrobial and say it again. Okay. They're produced by the mitochondria in perfect pairs, and then they are eliminated by certain enzymes and antioxidants like glutathione, peroxidase, superoxide dismutase, etc., that are already in our cells. And the trick, or the understanding of how these work inside the body, really depends on the balance between these different components. There's the smoke, or the oxidants, and then there's reductive also species. They're produced in equal forms by the mitochondria. Now that's what I was talking about before in the bottle of ASEA, that they're perfectly balanced. Not necessarily 8 and 8, but they're balanced. In such a way that they can be eliminated before they get into the nuclear DNA and cause damage or cause damage inside the cell. Okay? As long as they are in perfect balance, then they are produced at the same rate as they are destroyed. They're in homeostatic balance. The cell is healthy and well. If I were to take away the oxidants, if I took away all these red triangles here, the cell would be extremely sick and would not be able to right itself until we replace the red triangles. The real thing is that you need balance in your cells. You need this redox balance. When the redox balance becomes disturbed, that is when the smoke signals, as I call them, start to take place. Now, let me explain a little bit what this means. Suppose your cell is a house. You're sitting inside your house, looking at your fireplace, nice and warm, reading your paper, and all of a sudden you smell smoke. What would you do? Yeah. Well, turn to the sports section, maybe? No, you would actually be alerted. You probably go around the house or look at your fireplace and notice that, well, there's smoke pouring out of it. There's something wrong. I need to get something done, maybe you'd notice that the curtains have caught on fire or something worse. And when that happens, you probably will suspend every activity that you've thought about doing and just try to work the problem. The cells do exactly the same thing. When there's an imbalance, there's a buildup of smoke, if you want to think of it, they suspend their activities and they go around trying to find what the problem is inside your cell. Well, if they're able to find that problem, then they can fix it. If they can't fix it in about 2 hours, the fire marshals say, that's it, take it down. And the cell is then destroyed and digested. And a new cell then divides to take its place. That is healing on the cellular level, if you've been paying attention. That's how the cells know to heal themselves. We cannot take that process away from us and be healthy. In fact, that process actually is one of the primary processes that induce health in our body. Like you said, does it have something to do with the cell? If it does, then these redox signaling molecules are essential. Now suppose I've drawn this now to show you, this is what the body sees. It sees balance, 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 imbalance. Oh, something is wrong there. It'll send out messengers and then either fix it or destroy it, and then normal healthy cells will take its place. I mentioned that before. There are conditions, however, in which these oxidants build up and the damage is not able to be repaired immediately. It happens also because you age. Aged tissues do not repair themselves as fast. Immune system slows down, whatever. And you can't repair it as quickly. Now, what would happen if we were able to take our perfectly balanced mixture of these redox signaling molecules and infuse them into these tissues? I'd really like to know the answer to this question. I think that now we have a 200,000 person case study of people who've been taking this for many years, and we have seen things that are just incredible. And one of by the way, just on a personal level, I'm here today not because the science is so compelling, but I'm here today because maybe a few of you will start understanding how important this is and go and spread the word. Well, these cellular signaling molecules, you can see them under a microscope. We have a green dye that we can put in there, and we can see the reactive oxygen species in there, the ROS. And when we see damaged cells, malfunction of DNA, bacterial, whatever causes the damage, creates a buildup of these redox signaling molecules. You can see this under a microscope. It's kind of fun. And then this calls in the immune system to take care of the problems. People like this for some reason. Oh yeah, tank, I get it. Oh well, I don't know. But these oxidants also are very important. They're the immune system's choice of weapon of choice. They're also redox signaling molecules, part of this group. So the immune system then, when it has finished its job, retreats, and there are cells that have been damaged and destroyed, and normal cellular division then goes and fills in the holes. So you get the idea. Again, emphasizing that this is the process by which healing takes place on a cellular level. So what is ASEA? Here's my response. Okay, this is a scientist. The marketing people can tell you. In 3 words, it's a cellular performance optimizer. Basically, it just gives your cells the signaling molecules they need to function correctly. It works on the cellular level. It's used in energy production, damage detection. It's in the immune response, much more. It contains a perfectly balanced set, by the way, that's not 8 and 8, sorry, of the same redox signaling molecules that's produced inside the cells, and it's perfectly safe for plants, animals, and humans. So there you go. And that's what ASEA is. What are the benefits? Yeah. Oh, thank you. By the way, I don't get such a reception from the ASEA marketing department when I mention these. There will be people like Denny who will teach you how to say this message in a concise, easy way that it's understandable. Don't try to duplicate my message, okay? All right. The effect of redox signaling molecules on cell cultures. I'm going to go into studies now that we've done, and this is part of the road of discovery. We take cell cultures that exist inside our bodies and put them in a dish, and there we can grow them. Believe it or not, cells grow just perfectly well in a dish, human cells. And then we can put ASEA inside there to see what happens. And these are some of the studies that we've done. I'm going to go through these quickly. We looked at the translocation of some indicators that show toxicity. The people that did this are in national laboratories here in Washington, PNNL, National Laboratories, senior researcher. And he found that these cells exhibit absolutely no toxic response even when you replace 20% of the plasma that surrounds them with ASEA, which is much more than you'd ever get inside your blood. So, you know, we didn't believe that. So we looked at it another way, and we found the same thing. By the way, this is a toxic response. This is a normal cell. We used hydrogen peroxide, which is actually one of the components of this, to be our positive control. And this just gets more and more into the crazy as far as the science goes. We don't understand this still. But then we used these indicators of antioxidant production inside living cells. This is one of them. And we found that after 120 minutes in certain type of cells, even very small concentrations started to push the genetic buttons that produced antioxidants inside our cells. You can see this is salt water, and this is ASEA. And also these were activated by toxic in salt. So that's it. Anyway, I learned how to tweet, by the way. Not good at it. So anyway, nuclear translocation of these things. Thank you very much. So what does ASEA do inside cells? It increases antioxidant efficacy by, as you mentioned, up to 5 to 10 times. It helps cells recognize and fix damage. It increases the chances that healthy cells live and the damaged cells die. Let me explain this a little bit. We decided that we were going to damage these cells by X-rays, and we damaged the cells and put ASEA in there. And you know what I expected? Cells all recover, you know. No, half of them died. The damaged cells died, and then the normal healthy cells then exhibited absolutely no change. And so it seems like the ASEA actually helps to increase the chances that damaged cells die. That's actually an important part of healing, because what the damaged cells need to be able to die and then be replaced by healthy normal cells. Okay. Wow. So it also increases the efficacy of cell signaling. We put certain hormones inside these cell cultures and found out that the hormones are more effective. Here's something interesting. We had a lot of very aged cells. We call them senescent cells. They've been around in the laboratory for a long period of time. They don't do anything. They don't respond to hormones. They don't do anything, practically. It's also what happens inside our body when our cells start to age. we took these aged cells and poured in some ASEA, and all of a sudden they started to behave like normal, healthy cells again. So right at this point of the time, my heart was beating out of my chest. It renews the cell vitality. That's one of the, I guess, what we could say about that. Cellular antioxidants, these mailboxes which allow hormones and other messengers to get in and out of the cell, and the cell phones are enhanced by correct balance of redox signaling molecules. It increases the carrier reception of our cell phones. People can understand that a little bit. You know, people ask me, well, does it boost or enhance the immune system? I say, well, does having a cell phone call the fire department? No, not unless you need to. And then it's very handy to have a really good cell phone. And so creating better communication efficiencies doesn't boost really anything, but it helps the efficiency of the process when it's happening inside your body. Does that make sense? All right. So these signaling molecules mediate cellular communications and do a lot of other types of things inside of our cells. They make our cell more efficient, somewhat like oil makes our automobile more efficient. You really don't need to know all of the parts that it affects, but when you put it inside, it makes it more efficient. Also, has anybody done this? Have they changed the oil in their car and said, wow, my car is so much better now? We realize that, you know, the processes of changing the oil won't possibly yield immediate results, but they're absolutely essential for the maintenance of proper function. And it's the same thing with ASEA. It helps maintain proper function. So we put them in rats and then mice and a lot of other things. We have 20 years of data in mice, rabbits, dogs. And there's zero toxicity again. Mice ran 29% longer. Antioxidant capacities increased. Glutathione ratios, reduced glutathione ratios increased. Metabolism was normalized in these animals. Normalized meaning that they're able to utilize fuels more effectively. No endotoxicity, no lethal dose, no abnormal histopathologies. I throw this one in for the scientists. And you can see this science in the science page. ASEA will do a much better job at leading you down the path of discovery, as I've said, I think, as it should, on the website in the future. Results of animal studies. ASEA made mice run longer. They made energy more available to these animals. It increased antioxidant efficacies, and there again we saw zero adverse effects, even with large amounts. So now there's 20 years of studies. They all are saying the same things. Oxygen, nutrition, hydration, energy, and ASEA. Those are some of the most important elements in supplementing humans. So now what happens when we put these elements inside humans? Now I almost have to apologize because we looked at the metabolism first, because of course that is the process that creates these molecules, so we want to see what happens when these molecules are replaced inside our metabolism. We found that normal saltwater control people gave us a profile, a metabolic profile of something that looks like this. When we put ASEA there, that metabolic profile shifted. In fact, it shifted toward more efficient energy utilization as we think. There's so many different factors in here, there's no way that we can figure them all out, and if I stood here and I explained them all to you, you'd be more sleepy than you already are. But anyway, these metabolites caused these shifts. Then we were able to measure what they do inside the blood as far as the effectiveness on these metabolites. We found that after 30 minutes of taking 4 ounces of ASEA, metabolites were shifted. 90 minutes, another profile. And they kind of shifted and changed over time. The interesting thing is that up to 24 hours, we still saw the effect of ASEA in these people. So ASEA actually is effective up to even 24 hours after you take it. We're going to have a Q&A session after this.
What is PK?
Pharmacokinetic, I'm sorry. That's a special word that means that you're studying the activity of a certain substance in your blood to see if it gets to the right places in the body. So we can measure these shifts as an indicator as whether the ASEA product is getting inside the body and whether it's making changes to our body. And yes, we found, if you to look at this, just remember, ASEA is clinically proven to be able to affect things inside your body. It is getting in. So results of the human studies are that it's absorbed into tissues, it increases antioxidant efficacy. Again, it shifts the metabolites, and that's pretty much all we know at this point, and there will be more knowledge to come out in the future. But let me just mention one thing. I'm running out of time. Nobody has even stood up and yelled at me yet. Oh, well, anybody want to have a joke? All right. No, I'm sorry. I mentioned before that I was worried that my career would be over. And you kind of laugh. It wasn't such a laughing matter for me. But the reason being is that when we're dealing with new cutting-edge science, often all of these new discoveries are not part of the established double-blind placebo effect science that has gone on for 10, 20, 30 years and has been in scientific debate in these circles. What you're looking at are new discoveries that over time will be vetted out, and possibly then much of that will be included in the established science. James Watson, when he published his article saying that ROS molecules do things inside the body that are beneficial, actually kind of stepped out on a limb. He said, this is my hypothesis. I think these molecules are some of the greatest things that we will ever see as far as helping us overcome problems with our health. And he was willing to step forward. That sort of speaks to the importance of these types of investigations. In a little while, James Watson hopefully will be satisfied, and this will become part of the established science. It'll take billions of dollars and many years before we're able to see the whole picture. But right now, as I've explained to you, I don't need to see the whole picture to be able to know what the potential of this product is in the lives of people. You see people whose lives, their quality of life has been completely renovated because their cells started to act as they normally would and started doing what they normally should do. And that's the most beautiful mechanism of action that I can ever think. Most elegant and beautiful mechanism to help the body restore its own functionality. There's nothing else like this. So what can you expect when you start taking ASEA inside your own body? Well, I can't tell you. Everybody has a very different type of physiology. Everybody's a little bit different. And they will react to things in different ways. Everybody has a different level of redox signaling in their body. As long as it's balanced, the body doesn't care. If it's imbalanced, the body does care. It doesn't matter if it's balanced down here or balanced up here. You really don't know. maybe there has been damage in certain areas of your body that has been dormant for years, and it starts working there. There's no way to really know what is going to happen inside a human body. But you certainly can take it, and it's safe. And so you may get, for example, sleep quality, energy, mental clarity, aches and pains, recovery from exercise, and some other type of things that you'll start to immediately see. That's an indication that your body's starting to repair itself, and it's doing it more efficiently. So you can expect a kind of a replenishment phase when the body is readjusting to this optimal level of performance. And this readjustment phase then might take, who knows, sometimes 3 days, 3 weeks. But the body, I've mentioned this before, when it gets more energy and it wants to recover, it starts pushing garbage out. Just as if you were in your house and you said, oh, it's spring. Let me go and do some cleaning. Your house is not definitely going to get cleaner immediately. In fact, all the garbage that's around has to be pushed out, and then you can start noticing a difference. But the same thing with the cells. So as you start becoming more efficient, you'll push some garbage out. Please drink water. Copious and plentiful amounts of water when you're in this stage. The more the better. And I say about 10 to 12 glasses of water a day. Other people have different formulas. But, that's the way to get it out. Also, eating correctly. By the way, getting the correct nutrition inside your body is absolutely essential to this recovery process. It's essential. And athletes can tell you this. You don't perform, your body does not perform unless you have proper nutrition. And so, together with proper nutrition and hydration, especially because now the body needs more nutrition as it starts the repair process. It needs more energy. Give it to it. Let it happen, and then watch the results. But water and food are necessary then during this replenishment phase. Often then things will just kind of your body will accept it. Things will then try to level out a little bit. If you are having trouble, if you're miserable, take less ASEA. You can slow this down a little bit so that it doesn't hurt you or it doesn't make you miserable. And it'll take a little bit longer, but that's the way it is. So the most effective way to take ASEA is outlined in the product usage guide. What the product usage guide says is that you evaluate yourself on different areas before you take ASEA and several weeks after you started taking ASEA, and you evaluate yourself and see where the product is helping you or beneficial to you. Does that make sense? And that's really the only way that we can tell you what ASEA is doing. I can say that on a theoretical basis, however, since these molecules intermediate every communication between every cell in your body, increasing proper redox balance is going to help promote repair and replacement of the nervous system, in the nervous system. We've seen very clear evidence of this. These are the endothelial cells that we studied in our lab. It helps the cardiovascular system repair and replace cells. The redox signaling system also helps repair and replace skeletal muscular system cells. And these guys are probably the reason why these athletes are standing up and saying, hey, I have recovery that I've never seen before. I can go and run an Ironman and then 2 days later run another Ironman, and I haven't killed myself. And you know what they mean by killing themselves? That means they've damaged so many of their cells that they can't get up in the morning and do it again. But if you are able to increase that efficacy of the cells, boy, you've done a lot. I started reading all of these papers, and in my mind there are several questions. One is, well, will ASEA even be digested and go into your blood and get into your system? Will it do anything? And will it do some of this stuff? Will it help cells regenerate and things like that? I was pondering that for several months. My wife, by the way, I didn't take ASEA for about, I shouldn't say this, for about 3 or 4 months until I convinced myself that it was safe. Now I run around, to my family and anything, oh, we got a bump or scrape here. See that? So anyway, my wife was cutting vegetables and she cut herself. It was a pretty deep cut, about 4 millimeters, and it was bleeding. She couldn't stop it. I tell this story not because my wife thinks I tell it because she thinks that everybody will think she's clumsy. No. I tell this story because it actually has helped several people. And as it was bleeding, I decided, aha. And I put a little cup of this on the table, and I said, would you please stick your finger inside of it? It wasn't even ASEA at that point, called ASEA, but it's the same stuff. She put her finger inside, and all of a sudden the bleeding stopped immediately. The blood did not coagulate, but it didn't fuse either, just kind of sat there in the substance. And a minute later, she started cutting vegetables again. She didn't wrap it or do anything else with it. And I saw healing take place in minutes that should have taken hours. And healing taking place in hours that should have taken days. And that was the point, my ASEA friends, when I realized this was real. So anyway, thank you very much. I'm going to mention just a few more things. I have a few more minutes. Redox signaling is fundamental to processes in all forms of life on Earth. Animals, plants, every form of life on Earth that has a cell, multicellular form of life mostly, has these redox signaling molecules inside them. And it's interesting. I'll tell you another story. I have some time. The hardest people to convince of the validity of these things, after having a PhD in physics and spending many months and years in studying this stuff, I go to my brother-in-law and I say, he had a certain condition, and I said, why don't you try this out? This might help you. And he said, oh, thanks, Gary. And he went off, he didn't want to take it, so he put it in a plant. It was dying. And then he spread it on his dog, which had a big bad rash on one side, and that he'd been trying to get rid of for a long time. He didn't see anything, so he didn't think anything, went to sleep. The next day, he got up and his plant had revived. And the nice pink skin on the dog, and he said, oh my goodness. but really, until you've seen things like this, I don't expect that you'd really ever become convinced. Some of the scientists, by the way, that I deal with, if you try to think of how difficult it is in the network marketing sphere, just think how difficult it is in the science sphere, where people will say, where are your studies? Where's your proof? And something like this. And you have to stand up there sometimes and try to let them know what the state of the research is. But several researchers that I've dealt with shrugged it off, and then I said, well, okay, I'll send you a case. You can take a look at it. And they said, sure. And almost inevitably, they'd call me back. Hey, I had a knee problem that has gone away. Could this possibly have any of it? I smoked for several years, and boy, the stuff that's coming out of me is incredible. And I say, well, now I got your ear.
Yeah.
And I think that maybe that just can give you a hint as to also overcoming doubt. The product itself is strong enough to carry itself, regardless of how we do it. but it requires personal interaction, I've found. And this is the way to do it. So now, as a scientist, I'm actually becoming more and more involved in the network marketing industry. But there's another reason. There's another thing, you know, when I think of network marketing, I think of big cars and people, you know, standing up saying you can make $1 billion in 2 days.
2 days?
Well, okay. That's kind of what it feels like here in these meetings. And I said, I do not want to have any part of that. But the people that became associated with the ASEA Corporation were the finest, most genuine, humble, caring people that I've ever met in my life. And I said, I want to be part of that group. And I hope as we grow big that culture will never be lost. Mm-hmm. We'll try. So, DNA repair, antioxidant regeneration, oxidation, inflammation, antimicrobial stress, immune response, protective enzyme production. All of these things and many more are on the hot list of this research. And this list then is expanding exponentially. We will see more medical discovery. This is destined, I believe, to fuel the greatest medical discoveries that we will see in our lifetime and our children's lifetime and our children's children's lifetime. It'll be great. So with that, I'm done. I'm grateful that you came. Thanks for coming on this beautiful day that you could have spent outside and listening to a scientist lecture you on redox signaling. But I really appreciate all that you do, and thank you very much.